Okay, good morning. Assalamu alaikum for everyone. So, last week we started mutations. I guess students who could not attend live would have watched the recordings, which was uploaded in the YouTube. And a small part in mutations is remaining, which is the chromosomal mutation part. So with the completion of this chromosome mutation, not much on this content, only three disorders are mentioned. And after completing those three disorders, I'll give a small rough summary, right, for the complete mutation part. And after that, we'll start gene technology, which is our last topic in lesson number or unit number 7, right? Okay, I hope you have watched the recordings. So uh, let's go to the chromosomal mutation part, right? Chromosomal mutation. So last week we studied there are two types of mutation, gene mutation, chromosome mutation. And under gene mutation, we studied there were two types of disorders, right? Which were color blindness and sickle cell anemia. I explained that, and now we are coming on to the chromosomal mutation part. Chromosomal mutations bring about drastic changes in about genetic material and chromosomal structure, leading to abortions of fetus in the mammals. So, as I said, compared to gene mutation, chromosome mutation bring drastic changes, right? And what I said, a small point. Can you remember? Gene mutations bring minor changes compared to chromosomal mutation. Yeah, okay. But in gene mutations also, there is a type of mutation which brings drastic changes. What is that type? Can you remember? Can you remember nonsense mutation, which comes under substitution part, right? That nonsense mutation brings about drastic changes in the genetic material, right? So, can you remember this, right? Nonsense mutation, and chromosomal mutation and insertion deletion. These three bring drastic changes. What are they? Insertion and deletion of nucleotides, nonsense mutation comes under substitution, and chromosomal mutation. These three types of mutation brings about drastic changes in the genetic material. Okay, the remaining ones, what are they? Silent mutation and missense mutation. These bring about, compared to these three, they have minor changes, right? Okay. And they are saying these chromosomal mutations leads to abortions. Now we studied under chromosomal mutation the introduction which they gave under chromosomal mutation. See, I'll if you can't remember, I'll get that part again. Okay. See here, chromosomal mutation. Spontaneous abortions and miscarriages in mammals occur due to unusual numbers and structure of chromosome. Can you see here? They have already mentioned this point, right? They're saying spontaneous abortions and miscarriages. These two take place due to chromosomal mutation, unusual number or unusual structure of chromosome, right? And this is the same statement which are replaced or which is again they mentioned it under chromosomal mutation part. See here. They say chromosomal structures leads to abortions, drastic changes in genetic material, and chromosome mutation leads to abortions. Right? Can't see the screen. You can see what I'm writing, right? These red color parts, inserting deleted nonsense chromosomal mutation part. Can you really see that? Can see okay, okay. Okay. Second statement. If they survive, they show a peculiar set of abnormal characteristics in the phenotype which are called as syndromes. This is a definition. I have said you that start your collection of or compilation of your definitions. Right now, Unit 7, you are coming across large number of definitions. So start compiling them in a small book and maintain it. Right. If you separate it for each unit, it is more preferable. If you're doing for unit 7, start your compilation. Right now, we are coming across large number of definitions. uh, this will be more important, or this will be worthy during the latter part of your A level journey. When it is 1 month or 2 months for A level, this is very useful, right? Okay. What this statement says is they're saying if now normal abortions means, you can understand, right? Abortion means the premature termination of the fetus before it birth, it dies, right? In its fetal stage, right? Simply, that is the idea. Now they are saying if a fetus survives that abortion without dying, if it survives, still they will be showing an abnormal characteristic set of abnormal characteristics in their phenotype. Phenotype means the external appearances, which are called syndromes. Right? This is the definition for syndrome. There are three types of genetic disorders due to aneuploidy. You can remember aneuploidy, right? You have come across this word aneuploidy. So if you can't remember, I'll give a small information about that. Or again, I'll recall it. Right? Aneuploidy, and these are the few days you are coming across these words. Now just you have been introduced to these words, it won't be, quickly you can't recall them. So I'll remember that, right? Remind you that aneuploidy means condition where chromosome one chromosome is more or less. Simply, what is the condition? Plus one or minus one, that is the condition, right? Okay. Down syndrome. So they're saying these three genetic disorders are related to aneuploidy condition. They have one chromosome more or one chromosome less, right? Okay. The first disorder we are coming across is Down syndrome. Down syndrome is also known as trisomy 21 because cells of the affected individual have an extra copy of chromosome 1. Now extra copy of chromosome. Okay, extra copy of chromosome means one plus or one chromosome more or one chromosome less. What is the condition? More, exactly. So under aneuploidy, we studied two conditions. Can you remember? Something called monosomy and something called trisomy. Can you remember? Right? If you can you remember that? Now, trisomy means I said it is a condition. See, just remember this relationship. Monosomy means 2n minus 1. If you ask how does this relationship come, I'll explain that also. Right? Now, monosomy, what happens is monosomy. Just think a gamete. There is a cell. This gamete has been affected. Now, normally I said that normally during a cell division, there should be separation should take place equally during the anaphase. We know that from cell division, metaphase 1 and metaphase 2. During those, they will be arranged equally, equidistantly in the metaphase plate, and during anaphase 1 and anaphase 2, they will be separated. During anaphase 1, what happens? The homologous chromosome separates. During the anaphase 2, what happens? The chromosome separates at the centromere, separated into each sister chromatid. That is the scene. Now, actually, what happens here is they take place due to random arrangement. But if now aneuploid is related to errors, right? Now, just think there is a cell that has been affected with errors. Due to the abnormal arrangement, due to these errors, the chromosomes will be abnormally arranged, right? If they are abnormally arranged, their separation would not take place equally. Now, if one side gets chromosome, both of the chromosome, other side will be getting no chromosome. So here, one chromosome more. Here, one chromosome less, right? So this type of condition is one that is called aneuploidy, right? And undiploidy, they introduce us to two words called monosomy and trisomy. And what are they? Now, what happens here is monosomy means a condition where a gamete. Now, think this is a gamete which has one less chromosome. One less chromosome. So, normally, the gametes are haploid, right? So, if you consider gametes haploid, what is the relationship that is given in for normal gamete? So this gamete has one chromosome less. That means how the relationship will be coming. If it has only one chromosome less, just for example, right? Now, for example, just think this is a gamete and it has one chromosome less than the normal haploid level. What is the relationship? What type of relationship? N minus one, exactly. Right? N minus one. And now there is egg. This is sperm's egg. This is a normal gamete. So it has an N. Right? Now we know there is a fertilization taking place. And after fertilizing, what happens? They form a zygote. Right? They'll be forming a zygote. So normal zygotes will be having 2N. Right? Why? Normal gamete will be having N plus N. It will become 2N. But here it is N minus 1 plus N. So normal maths, just general maths. Let's see. N minus 1 plus 1. So both mass plus and plus, right? So 2n and then minus 1. This is the relationship, right? This is the relationship which will be coming. Can you understand? Am I fast? We have discussed this last week, but still, as a revision, I'm sorting those information into a small uh, diagram. Is it clear? Clear, right? Okay. Perfect, perfect. Right. Now, if you see here 2n minus 1, see here, this is what I said. Monosomy is 2n minus 1. So monosomy means simply what is the condition? One chromosome less. So if you know monosomy, you can just assume or you can just have a guess of trisomic condition. So here one chromosome less, right? So if it trisomic condition, this gamete will be 1 n plus 1 and this gamete will be n. So normal gamete fusing with a abnormal trisomic gamete. This is a trisomic one. So this is a monosomic one, right? So if this fuse, this forms zygote which has 2n plus 1. So that is a trisomic condition. Right? Okay. Now here what under Down syndrome what they are saying is Down syndrome is also trisomy 21. Just highlight that word trisomy 21. Trisomy means it depicts one chromosome more. Okay. 21. What does this 21 mean actually? If you ask what that means, this is what they are meaning. Now if you see every class I'm used to mention this, right? Just to make you practice to this information. I have said that each body of our human body contains how many pairs of chromosome? 23 pairs of chromosome. In this 23 pairs, 22 pairs of chromosomes are what type of chromosomes? Can you remember? 22 pairs of chromosome. Autosomal, very good, very good. They are autosomal chromosome, right? That remaining one pair is said to be what chromosome? What type of chromosome? Sex chromosome. Very good, very good. Right, okay. Now here, first 22 pairs belongs to autosomal chromosome. Okay. Now here if you see Down syndrome is also trisomy 21 because the cells of the affected individual have an extra copy of chromosome with 21. In the chromosome 21. Now it contains first 22, right? 21st chromosome. This 21st chromosome has an extra copy, which means n plus 1. This is what they try to convey. Is it clear? Is it clear? Yes. Is it clear? For all? Okay. Okay. The syndrome shows characteristic facial features, short body, heart defects which can be corrected, and developmental delays. These are some type of characteristics that have been shown by them. They have characteristic facial features, right? uh, Short body. They have a short height, heart defects that are present, but these can be corrected. It seems developmental delays. That means normally, if you see now a 19 years child, right? He will be growing. Just think he's growing for 6 feet, right? These ones who have been affected with Down syndrome, they'll be showing 4 feet or 3.5 feet. So that will be developmental delays, not the actual developmental state for their actual age, right? There's a limit, right? For this age, a person should have this much of height and this much of weight if he is normal. But these people will be under that limit. They have a high risk of developing what type of diseases? Leukemia and Alzheimer's disease. Leukemia, you haven't come across leukemia, okay? But you have come across Alzheimer. Can you remember where we came across this disease? Alzheimer. In which lesson? Can you remember? Nervous system, Alzheimer's disease, right? There we come across this Alzheimer's disease. Almost all males and half of the females with Down syndrome are sexually underdeveloped and sterile. Their lifespan is shorter than normal but can live past the middle age with proper medical treatment. Okay. They have a high risk of developing leukemia and Alzheimer's disease. Almost all males and half of the females are being affected with this Down syndrome disease and they're sexually underdeveloped, which means sterile. They can't reproduce like normal humans. These have uh, sexually underdeveloped characteristics, right? However, they have a low risk of high blood pressure, atherosclerosis, which is the hardening of arteries, strokes, and many type of solid tumors. Now, see, this is a type of benefit which these Down syndrome patients have. They have a low risk, right? Compared to that, this is a disadvantage. Yeah, they have a high risk. Sorry, not this one. Wait. Not this one. This one. Yeah, this one. They have high risk of developing leukemia, Alzheimer's disease. So this is a benefit and this is a disadvantage. Can you see? All right. Okay. Despite the abnormalities, they may live independent and employed. The risk of having a baby with Down syndrome increases with the age of mother. This is caused by non-disjunction. We came across this word last week. If you can't remember, no issue. I'll again repeat that. What type of information I gave last week about non-disjunction. Non-disjunction means the inability of separating chromosomes during meiosis, right? Now I said that, yes, I said that normally the cell during its anaphase they should separate, but due to an error, due to an abnormal arrangement, they won't be separating. That inability to separate is called non-disjunction. Simply that is the idea, right? Is it clear? No doubts, right? As this is the first time you are coming across these words, you might forget these words easily. Is it clear what is non-disjunction means? Clear. Perfect. Okay. What they are saying, the risk of having a baby with Down syndrome increases with the age of mother. A baby which is born with this disorder, the risk of a baby born with this disorder increases with the age of mother. Now it is a indirect statement. They are saying when the mother's age, think the mother's age increases. When the mother's age increases, what happens? The the probability. Let's use the word probability of non-disjunction taking place also increases. So non-disjunction means the inability to separate the chromosome. So if this take place, what happens? There would be an abnormal separation taking place. So if abnormal separation taking place, they will be leading to what type of condition? There's a specific word. I want the word from you all. If you understood this concept, you can tell that word. What is that? With the increase in the mother's age, the probability of non-disjunction taking place during their cell division increases. If the probability of non-disjunction increases, there is a condition that leads. What is that condition? What is that condition? Abnormal separation leads to what? Some sort of condition. What is that condition? This leads to aneuploidy, right? Aneuploidy state. So mother's age increases. Exactly. So probability of non-disjunction increases. So at this, due to this increase in that non-disjunction, aneuploidy take place. The aneuploidy, one chromosome more or one chromosome less takes place. Right? Okay. So they're saying the risk of baby born with the syndrome. So after a mother has been affected with this aneuploidy condition, the baby that is born for them also contains the same condition. That is what they're trying to convey there. So when the mother's age surpasses a limit, and after that passing that limit, if they are conceiving for a baby, which means they have increased their probability of or chance of non-disjunction in their body. So if that take place, their body cells will be leading to a condition called aneuploidy state, where one chromosome more, one chromosome less is present. Why? Due to the abnormal arrangement in chromosome, right? So if this is the case, the child born will also have a risk. I'm not saying it will be compulsorily having. I'm saying there is a risk where the baby can be affected with the Down syndrome condition, right? Okay. While Down syndrome is due to trisomy of autosome. Now, by seeing these two words, can you remember the relationship which I gave? If you can remember, then you have understood the concept. While Down syndrome is a trisomy of an autosome. See, can you understand that? If it is hard, let me know. Any difficulties in understanding that? Now, if you see trisomy means just remember this relationship 2n plus 1. Autosome means any chromosome less than the 22nd chromosome, including the 22nd. Right? Okay, let's see here what happens. I said that it is affecting the 21st chromosome. So it is belonging to what type of chromosome? Autosomal chromosome. And it is a trisomic condition, it seems. Correct, right? Why? See here, the first statement itself says that it has an extra copy, which means it is a type of trisomic condition. Right? Okay, they're saying Down syndrome is due to trisomy of an autosome. Right? An autosomal chromosome has a extra chromosome. There are human genetic disorders due to aneuploidy in sex chromosome. Aneuploidy in sex chromosome means, see here, in sex chromosome, aneuploidy condition take place. Aneuploidy condition can be n plus 1 or n minus 1. Any of them. Alright. The aneuploidy condition in sex chromosome causes Turner syndrome due to monosomy, Klinefelter due to trisomy. Ah, simply this is the idea. Monosomy. Wait, it seems to be jumble, right? See, you see the sex chromosome. How many sex chromosome pairs are present? One pair. So if you consider this one pair, they are saying there are disorders that can take place where sex chromosomes are related to aneuploidy condition. Aneuploidy condition contains two possibilities. One is n plus 1, which means one chromosome more, n minus 1, which means one chromosome less. Right? And they are given examples for these conditions too. N plus 1 and N minus 1. N plus 1 means Klinefelter, it seems. It is a disorder. Right? N minus 1 means Turner syndrome, it seems. It is also a disorder. Is it clear? Now, the Down syndrome, which we learned now few minutes ago, was a disorder that is affected with autosomal chromosome. Right? The 22 pairs, remaining 22 pairs autosomal chromosome. Uh, Now, Down syndrome is related to this. Now, these two diseases, where we are going to now learn about them, these are related to sex chromosome. Is it clear? Is it clear? Clear, right? Clear. Very good. Very good. Okay. If you have any doubts, let me know. No, don't be afraid to mention your doubts. Right? If you have any doubts, not your mistake. It's my mistake. I have you know failed to explain that to you. Right? If you have any doubts, drop a message. If it's even the first part, I'll again go and explain it again. Right? Don't you know uh, panic to ask any doubts. Right? Okay. Now let's go to the points. Right? This is clear, right? The last point, which says Turner syndrome is due to monosomy. Let's see, is it due to monosomy? Turner syndrome, monosomy. Correct, right? Monosomy means one chromosome less, n minus one, right? Clean filter due to trisomy. Clean filter due to trisomy. Trisomy means n plus one chromosome more, right? Okay, so this part is clear. Then Down syndrome is clear. See, next part is Turner syndrome. So just go through this Down syndrome note and let me know if there are any doubts. If no doubts, let's move on. Clear? Is it clear for all? Clear, right? Okay. Then let's move to the next part. This is Turner syndrome, right? So as they mentioned it at first, right? Turner syndrome is due to a monosomic condition. Monosomy in X chromosome. Turner syndrome is due to monosomy. Monosomy means one less chromosome. That's the simple idea, right? Monosomy. Mono means one, somy, chromosome. So one chromosome less than the normal haploid condition in X chromosome, right? In very rare cases, there are females with only one chromosome, and hence their genotype is XO, and this is called as viable monosomy. So normally we know females have what type of genotype? XX. Males have XY, right? In these conditions, if female is affected with Turner syndrome, there are conditions where they have one chromosome less, one X chromosome less, which means X. So actually it will be looking like X, right? But still it is like a number of seat. Now just think, this is a seat, right? This is another seat, right? Now there are two seats. Now there are two seats. There was a passenger here. There was a passenger here. Just think these are X chromosome. Now if one chromosome is less, how will they look like? They're looking like this. One chromosome will be present here, other will be empty, right? We're looking like this actually. There's no space. But as there is a location, right? Can you understand? As there is a specific location where a chromosome is present, there is empty, right? So instead of just putting it as X, they are mentioning it as XO, just to make sure that there are two alleles. Is it clear? X, X. Just to make sure there are two alleles. Two alleles means two versions of genes. Now, simply this is the idea. Now, if you can't, if you find it difficult to understand, let's go to the previous part. See here. Wait. This would be helping you. Wait. This part. See here. We learned at a color blindness, right? Now with color blindness, see here. One more. One. See here. Next part. In females at heterozygous condition, a defective allele in one chromosome is masked by the good allele in the next, which means we under color blindness we studied that color blindness will be affecting what type of chromosomes? X chromosome. So females have how many X chromosome? Two X chromosome. Males have only one X chromosome, right? Now, if this if X chromosome is affected, their phenotype will be completely affected by there is no any source of X chromosomes because males have only one X chromosomes. As they have only one, they should protect it. If it is affected, then completely characteristics that are controlled by this chromosome will be affected. But if in this case, now if this is affected and if this is good, this will be masking the effect. Now like this is the good one, and this is the affected one. It will be masking the effect. It will be coming in front of it and just like masking it. It will not be expressing its effect, right? Now, can you see these are two alleles? That is what they convey here. See, in female heterozygous condition, defective allele. See that allele in one X chromosome is masked by the good allele in the other. Is it clear? No? Now this is the thing. Clear? Right. Still, I'll mention this one. Now, if you see on the first class itself, I said that just think chromosome. This is a chromosome. In the chromosome, I said that there are some band-like structures like this. What are they? What is this in chromosomes? What are these band-like structures called? Can I remember? No? They are called genes. Right. Now, for genes, I said that instead of remembering as genes, you can remember it as segment of DNA too. Segments of DNA. Right? If you closely look that, it will be looking like this. Adenine, guanine, cytosine, adenine, thymine. Right? It will be looking like this. Right? Now, if you see genes, I said that genes control characters. Right? If genes control characters, always there is a alternating version. So you might ask, sir, how is this alternating version present? Simply now see, uh, Can you remember I gave an introduction or some sort of information about homologous chromosomes? Now you might feel, sir, don't go in deep. We can understand, but still, I want to make sure you completely understand this. Just partially understanding it, when you come across these again, or when you are revising it again, you will feel difficulty in understanding that. So I don't want that to happen. Right? I'll explain it again. Try to understand. Right? We have plenty of time. We can complete this before today, and hopefully I'll complete one or two topics in gene technology too. Right? See here. Now this is, sorry. This is a chromosome. This is from the father. This is a chromosome. This is from mother. Right? Now just think there is a gene. This is a gene. It is located 1 nanometer from this top part. And this gene depicts skin color. See the word depicts, right? Skin color. Just think this is black. Now, if you see, actually, if you measure here, or yeah, we won't be measuring it, but actually, if it is located at the same location, same 1 nanometer distance, if it is located, if there is a gene which also depicts the skin color, which also depicts the same characteristic feature, skin color, these two chromosomes are called what type of chromosomes? Can you remember what type of chromosomes? Ah, exactly. Very good, very good. Homologous. Okay, now here you should have a clarification that I'm not speaking about the trait. I'm speaking about the characteristic. Now it should depict the same characteristic feature, skin color, but the trait can differ. If this is black, this can be white. Now, if you see, now if these fuses during fertilization, chromosomes and 23 chromosomes from mother, 23 chromosomes from father will be coming, right, and it will be forming a zygote, right? Now, zygote will be containing these two, one from father, one from mother. This depicts black color, which is from father. This depicts white color, which is from mother. Now, can you see there is a chromosome which depicts the skin color, and there are two variations, one black or one white. Can you see? Can you see there are two variations? One is black, one is white. Now, this is the zygote. Now, this is for zygote, or this is for the child. They both will be given to the child, right? So the child contains one chromosome, skin color depicting one contains one variation, can be white or black, but both are present. Can you see? Do you agree with that? No? Yes. I want both. I want all the students to understand. Ah, clear, clear, right? Okay, okay. Okay. Now you might ask, now there are two colors, black and white. What color will be depicted by the child? Will it be black or white? Ah, here only will be considering something called dominant recessive character. What type of character? Dominant and recessive character. Now here we'll be seeing which is dominant. If father's black gene or black allele is dominant, the child will be starting to show a black color in the skin color. If mother's white is dominant, it will be showing white color. So this the expression completely depends on dominant recessive character. I'm not going on to those topics. What I want here is that you should understand for every gene. Now see here, what is the gene we spoke about? The gene we spoke about was gene that depicts skin color. Just think skin gene. What was the gene? What was the gene? Students, what was the gene? Skin. Now, the gene that controls skin. Now, answer this question. What are the alleles? What are the alleles? Alleles means alternating versions, different variation, versions. We can use various words. Now, if the gene is skin color, if the gene is skin color, what are the alleles? Black skin, white skin. Very good, very good. You have understood that. Can you understand that? If gene is skin color, allele means black or white. Now see here. Now if you see like this, it is bit understanding, right? Now see, gene. Gene is skin color. It depicts. What is its versions? Its versions of a gene are called allele. That is the simple idea. Versions of a gene is called allele. So there is a proverb, right? Every coin has two sides. Every coin has two sides. So we, science students, we can say that every gene has two alleles. Is it clear? Is it clear? Yes. Okay. But still, there is a problem. Now, if you see father and mother both share the same allele, both of them show the same white allele, what will be the condition of the child? Child will contain both the alleles. It will be containing here white and here white. Now, here there will be two different alleles or one same allele. Two different alleles or one same allele. If both of them have the white skin allele. Same. So there are how many types of alleles are present? How many types? I'm not asking how many. I'm asking how many types of alleles. One. Very good. That's it. Is it clear, this part? Now, this is not related to your the syndrome part, right? I want you to give an idea on this. Clear, right? Complete part. Now, what I explained now, few minutes ago, this complete section, is it clear? If it's clear, I'll move on. Clear, right? Okay, perfect. Then let's move on to the note part. Where is it? Right. Turner syndrome is due to monosomy in X chromosome. In very rare cases, their females have only one X chromosome and his genotype is XO. Now you can understand, right? Now this. Wait. Okay. Right. See here. Now you can understand, right? Each of them, each of the gamete has XX for females, XY for males. Right. Now they are saying due to monosomic condition, one chromosome is lost. So what is the condition? N minus one. So if it takes place in females, they get a genotype. This is the actual genotype X, because why? Two X were present, one X was lost, so it is X. But they are saying as there is a location for the allele, there is a location. They are mentioning it as XO. Is it clear? Without just mentioning it as X. Why? Because all of them have two types, right? Instead of just mentioning it as X, they are mentioning it as XO. There is a vacant, but it's empty. Is it clear? Clear? Clear. Okay. Perfect. That is the hardest part. Then you can understand others. So this condition where XO is called viable monosomy. Viable monosomy. These individuals are phenotypically female, but they are sterile. Phenotypical means when we see out externally, They seem like females, normal females, but they are sterile. They can't reproduce because their sex organs do not mature. Organs do not mature. When girls affected with Turner syndrome are treated with what is the treatment? Estrogen replacement therapy. Why? Estrogen is one of the hormone which involves in inducing secondary sexual characteristics. Can you remember? Under reproduction, we'll be learning this. If you haven't learned it, no issue. Right? Estrogen is a hormone which induces the secondary sexual characteristic. Secondary sexual characteristics means growing taller, right? Enlargement in breasts, development of uterus. There are multiple characteristic features, right? So all of these in females as well. Now in females, it is mainly controlled by estrogen. In males, it is controlled by testosterone hormone, right? We'll be learning it under unit number 5, reproduction part, right? When girls affected with Turner syndrome are treated with estrogen replacement therapy, they develop secondary sexual characters. Okay, by inducing this treatment, they can develop these characters. They have a short stature. Short stature means height is short, short height. Some may have extra skin on the neck. Extra skin on the neck means normally we have a curve here, right? The neck. They have a complete solid part. Now, if you see, this is how it looks like. Now, a normal person has a head like this, and he looks like, no, this is not a tree, eh? It looks like this. It, see, it looks like this, right? Now, these affected individuals, they have some sort of neck like this. This curve is absent. Instead of this curve, there is skin, right? Can you understand? Is it clear? Clear, right? Not hard to understand that. Clear? Okay. Other characters are these are the characters shown. Puffiness or swelling, lymphedema on the hands and feet. Puffiness means now have you seen some diabetic patients have this condition? A puffiness due to some excess liquid. Lymphedema means excess lymph, right? What happens is they get swollen hands and legs, right? If you press, a mark can be seen. That much of swellen, right? So these Turner syndrome affected individuals show that characteristic feature. It seems skeletal abnormalities, skeletal system. There are certain effects, heart defects, high blood pressure, and kidney problems, right? Turner syndrome affected individuals have characters like this, right? And last one, most of them have normal intelligence. That means normal intelligence. Some people say that these individuals now the Turner syndrome. Now I don't know. Have they given a picture for the Turner syndrome in your resource book? Have they given any picture for Turner syndrome in your resource book? A woman's no? Haven't they given a picture of a woman? No. I see. No. Wait a minute. Wait. I am searching for a picture, but no issue. Right. Okay, okay. Let's back to the get back to the note part. I had a picture of it, but can't find it now. Right. Just go through this Turner syndrome part. Now, normal intelligence means most of the people think that due to this condition they will be affected mentally. Now, normal intelligence means they are not affected mentally, which means they have normal intelligence, right? Some people will be acting like they, we normally use the word mental, right? Uh, so these people are not so. They have a normal intelligence, which means they can understand, right? They know emotions. They have some norms, some feelings, so they can take certain decisions. So they have a normal intelligence, right? Mentally, they are stable. That is a simple idea. Right, just go through this Turner syndrome part and let me know if there are any doubts. Is it clear? Clear. Is it clear for all? Right. Now, when you come across these disorders, you might feel there are so many characters, there are so many symptoms, there are so many treatments, right? So, with practice, you will feel these are very easy, right? So, what we'll do is, I'm planning to, you know, practice you all in disorders, some sort of disorders, diseases, and their cause, what is their treatment, right? How they are caused. So these we'll be practicing them. So I have a plan. Now we have learned so many of diseases, right? From unit number two. Unit number two, can you remember the first disease you came across? The first disease. Can you remember? In your A level journey as a science student, the first disease you were introduced to, what was the disease? Cancer. In unit 2 on the cell division part, you learned under cancer. Can you remember? Cancer and galls. So galls will be present in plants, right? So for human body, the first disease is, which we learned was cancer. Am I right or wrong? I don't have the resource book here. I hope it was the first disease. Ah, right. And then onwards, we came across various diseases in Unit 5. In each system, digestive system, now see, digestive system had four, I guess. And then we came to respiratory system, circulatory system, nervous system, excretory system, immune system, hormone endocrine system. And then we went on to why type 1 diabetes mellitus, type 2 diabetes mellitus. Uh, and then we move on to reproduction, skeleton. So there is large number of content, which means now for each section, for each system, we had various number of diseases, right? So as students, it feels a bit hard to handle those now. However, in every year they will be asking from a disease, right? It can be in structure, it can be structure, or it can be an MCQ question, or it can be an essay question. But essay, it is very rare. So these two sections mostly they ask about disease and disorders, right? So what we'll do is we'll study for the time being. We'll study these, and what we'll do is very soon I'll be starting that practice with questions which are related to this disease and disorder. I'll give some sort of summaries and notes, right? So don't worry about that, right? Okay. So, and the last one for today's topic, which means under disorders, chromosomal mutations, is Klinefelter syndrome. This is due to a rare condition of an extra. So extra means trisomy or monosomy. Extra. Can't you see the tute? Wait. Now can you see? Yes. Okay. Sorry. Klinefelter syndrome. This is due to a rare condition having an extra X chromosome. So extra means, is it trisomy or monosomy? Trisomy. Very good. Very good. Trisomy. Right. So simply extra. So this TR trisomy. Just remember like that. In a geno with a genotype of XXY, since it carries a Y chromosome, these are individuals are male. So normally a normal male has XY, but here they have XXY, an extra X chromosome is present, right? But still, as they have Y chromosome, they are males. Phenotypical, they are males, but still they show some abnormal characteristics. Let's see what are they. Although these people have the male sex organs, they are sterile. See here, they are sterile. So all three disorders which we came across had this word sterile, which means they can't reproduce normally. They have the ability of reproducing, but compared to normal human, they are sterile, it seems, right? They are see the characteristics. Their testes are abnormally small. One out of two X chromosome. One out of two X chromosome is inactivated. Even though two X chromosomes are present, one is inactivated. But for the time being, there's just an X chromosome is present. Yet these males have enlarged breasts. Now see, enlarged breasts is one of the features shown in males or females. Males or females, normally. This characteristic feature is shown in normally males or females. Normally, continue. I was speaking about normal individuals, female. But here, this is present in male. Why? Because of this extra X chromosome. Can you understand? Can you understand? Yes. Now you might ask, sir, you said that there are two X chromosome and one Y chromosome, and they said that one is inactivated, but still, due to that inactivation, how come he shows some female characteristics? They have X chromosome, but still it is inactivated, right? You might ask that question, right? Right? Yes, you are right. There is two X chromosome, one is inactivated, but still, how come this characteristic is shown? Now, inactivated in the sense they are saying that if these two X chromosomes are present without any inactivation, he will be a actually 90% he would look like a female. Why? Two X chromosome and Y chromosome. Female and male characteristic will be present, both. But most of the time, he has a complete female genotype. So he'll be more likely to be a female. But as only one is inactivated, as the inactivated, still, as this is inactivated, he look like a male. But still, as there is an X chromosome, he will be showing certain female characteristic. This is the same. Now see, simply I understand, explain this in this manner. XXY, this is inactivated. As this is inactivated, there will be male phenotypically, but shows female characteristics, few female characteristics, right? Now, if this is not inactivated, what happens? He will be more likely to be a female, more likely to be a female, showing male characteristics. Can you understand? Why it is not like this? Because it is inactivated. Even though it is inactivated, still there is X chromosome, right? Even it has no effect, still an X chromosome is present in its genotype. As it is present, it will be showing certain few female characteristics. Is that point clear? Clear. Okay. They have a subnormal intelligence. Subnormal means, now here in Down syndrome we understood that normal intelligence, right? Subnormal intelligence means they have some sort of inability in understanding, right? They can't take individual decisions, right? Bold decisions cannot be taken. They seem to like they normally like to be now underaged people. Now if you see, some people are there who are 40 age, right? They are 40 years in age, but they will be behaving like 15, 16, right? Some are present, they are 80 grandfathers and grandmothers, but they behave like uh, 5 or 6 years. So similarly here also there is a specific age, but due to the subnormal intelligence in Klinefelter disease, they'll be behaving like underaged people. XYY trisomy will make males and XX trisomy will make females, which means XYY condition. Now, is it a trisomy or monosomy? XYY. Trisomy, right? So, XYY condition and XXX condition are normal, it seems. These two are normal. They do not show any syndrome and have normal female characteristics. They are fertile and slightly taller than average. Now, this is the thing. Now, here, under Klinefelter, we are speaking about a condition where XXY, right? This is abnormal. Why? In a male, there is extra X chromosome. But there is a condition in Klinefelter where XXX can be present or XYY can be present. If you see here, these two are normal. Why? Here an extra X chromosome is present. However, she is female, still there will be an extra female characteristic feature. However, he is male here, XY, extra female male characteristics can be present, right? But these two, the problem is they are bit height. The normal height, now for humans, normal height is this thing 6 feet, right? These people will be showing an extra height. They can be 7 or 8 feet. They are slightly taller. That's the only difference. Slightly taller, but they're fertile like us. Is that clear? Is that clear? Now all these three are trisomic conditions, right? XY is abnormal, right? Why? An extra X chromosome is present in a male genotype. XXX is normal. Why? They are already female. Having an extra X does not impact them much. They'll be showing same female characteristics. XY is normal, which is a male. Having an extra Y chromosome will be maintaining still the male characteristics. Is it clear? But the only problem is they are slightly taller than us. Is this clear? This part clear? Okay, perfect. So just go through this Klinefelter syndrome part and let me know, do you have any doubts here? Clear? Is it clear for all? Clear. Okay. Okay, then now finally there is a last part here. What is this genetic counseling? Right? Here, this complete part, right? This complete part they have asked, I guess, for the past 10 years. Yeah. For the past 10 or 15 years, they haven't asked a question in MCQs. Only once they asked a question in a structure paper, right? Uh, so what you can do is, if I try explaining all these, I don't think it will be effective. So what you can do is just leisurely, right? Don't try to memorize this part. Leisurely just read this part, genetic counseling part. Now in this unit 7 there are certain sections like this. Now this part and last part, which is Cartagena protocol, there is a protocol type note, right? These they have given in bulk of notes and they are expecting to just go through them, right? Not to memorize, you just go through them and leisurely see, are there any points that you can't understand, right? They won't be asking in detail what is genetic counseling and those stuff. They'll be just asking some information, right? I don't think they'll be asking generally. They haven't asked for the past 15 years. Only once they have asked in a structure paper. That's it, right? So just leisurely, just go through that, right? Or if you want to explain it, also I explained, which is better. Just going through, which is better. Is it okay? Right. Okay then. So finally, we are coming to the end of the mutation part. Right. Now I have a small work to do. What is it? What it is means now we have to do a small summary. Right, right, right. Now, even if you haven't uh, revised the previous class, what we'll be doing now is we'll be revising all the sections under mutation now. Right. So I want your help now. So I'll be asking certain questions. You just answer it. Right. Okay. First one. We started with mutation. Right. This is the summary. Yeah. A flow chart type. First, mutation, right? Mutations are how many type? Two types. First one is gene mutation and then chromosomal mutation, right? Normally, this mutation, wait. Rather than doing, rather than doing like this, what I'll do is I'll take a full page. That is easy, right? Right. Okay. Mutations. First, what we'll do is we'll start with the definition. What does the definition say? Mutation means alteration of nucleotide sequence of a genome of organism. This is the definition, right? Clear? And they're saying mutations normally have three types of impacts. What are they? They can cause three type of impacts. Can you remember? First one is lethal, second one neutral, third one beneficial. Right? And then we are coming to the classification of mutations. Right? What are the classification? Mutations are divided into two types. What are they? Gene. What are they? Gene mutation and chromosomal mutation. Wait. Right. Gene mutation and chromosomal mutation. Right. Now these mutations are classified based on some sort of aspect. What is that? Can you remember? Mutations are classified on the based on the the scale of alteration of genetic material. What does that mean? How the mutation alters the genetic material. Depending on that, they are divided into two: gene mutation and one is chromosomal mutation. Up to this part, do you agree with me? Now, this is what we have learned in the previous class. I'm just giving a rough idea. Clear? Shall we move on? Yes, perfect. Okay, let's come to the gene mutation part. Gene mutation. As usual, we'll start with the definition. Right? What does the definition say? The permanent permanent alteration of DNA sequence of gene of an individual. Right? Okay. Gene mutations are caused due to three main factors. Right? Let's see what are those factors. Wait. It's not straight. Right? It is classified or it is caused by three main factors. No. What I'll do is right. Wait. Ah, let's do like this. It is caused by three factors. What are they? Try to recall. What are they? First one is errors in DNA replication. First one is errors in DNA replication. Right? Second one, what is that? Second one is physical factors. Third one is chemical factors, right? If gene mutations are caused due to errors in DNA replication, what are they called? They are called some sort of mutation. What are they? Can anyone remember that one? They are called spontaneous mutation. They are called spontaneous mutation. So gene mutations which are caused due to errors in DNA replication, they are called spontaneous mutation. Okay, physical factors, chemical factors. Example physical factors. Yes, we have some examples. Let's see what are examples. Examples: UV rays and X rays. Chemicals normally carcinogens. So normally carcinogens are mutagens. So that is the main idea. Okay, now these three, what are they called generally? Errors in DNA replication, physical factors, chemical factors are generally called as mutagens. Mutagens means mutation causing agents. Right? These mutations means all three, yeah? All three are causing agents for mutation. Right. Until this portion, is it clear? Are there any doubts? Keep your tute near, right? Keep your tute or the resource book closer to you all and just check with the information. Now, I don't have a resource book here. So if there are any mistakes or if there are any doubts, let me know. Right? Is it clear? Clear. Okay. Then let's go on to the classification of gene mutation. So gene mutations are classified into three. What are they? First one is substitution. Second one, insertion. Nuclear pair, single nuclear pair substitution. So there's a full term, eh? A single nuclear pair substitution, nuclear pair addition, nuclear pair insertion, nuclear pair deletion. So these are the full terms for the rough. Now I'm giving a summary, right? So I can't be writing the complete statement, right? Insertion, and third one is deletion. These are the three types of mutations, gene mutations, right? Substitution. Substitution means simply for a simple idea, right? Let's put a bracket and write. It means replacing. Substituting means replacing a nucleotide base pair. Inserting means adding nucleotide base pair. Deletion means removing nucleotide base pair. Right? Okay. I don't know whether I can hit the notes below here. Let's see. So substitution, if you see, substitutions are classified into three. What are they? Can I remember? First one is silent. Second one is missense. Third one is nonsense. right? Silent, sorry, silent means substituting a single nucleotide pair, especially the third nucleotide. So normally the codon, see here, codons, right? Now if you see A, G, G, C, G, thymine, so uracil, thymine, uracil, sorry, let's use this as uracil, adenine, sorry, no, I'll use this one, right, wait, I'm using uracil. Now this is a codon, this is a codon, so triplets, right? This is what I say as third letter of a codon, right? So normally, when a third letter of a codon is replaced, the mutation caused is called silent mutation, which means there is no change in the final amino acid that is coded for. Is it clear? Okay, let's mention that one also. No change in the amino acid coded for. Right? There is no change in the amino acid that is coded for. Before the mutation, what was the amino acid? Same is the after the mutation. Right? Why? Why it is the reason? Because third letter in a codon is a wobble, which means changing it does not have a huge impact. Right? This is called a silent mutation. Right? Missense mutation. Missense means normally if you see here, this is the third, right? If I change this, there would be huge impact. Why? Now you see AGG. Use your that codon book and let me know what is the amino acid code for AGG. Drop a message. AGG. What is the amino acid coded? ARG. ARG. Right, this is the amino acid. Now AUG. I am replacing this G with U. AUG means methionine. Right? If AGU, I'm replacing this third letter with U. What is that? AGU. AGU. AGU means SCR. Right? Right. Now, if you see here, most of the time, right? If you see here, GGG. GGG means what is the amino acid I get? Oh, okay, GL. Now I'm replacing this GGG with a C, which means GGC. What is the amino acid I get? Same, right? See, changing the third letter gets the same, which means it is a wobble. Changing it same, changing the third letter will be getting the same amino acid most of the time, right? So there are chances where it can be not the same. See, here is one of the chance where here AGG, I changed this G with U. I got SCR. Can you agree that? So that statement is important. Normally, change the third letter, say remains the same. If it changes, the amino acids coded for that type is called what type? Missense, which means changing a nucleotide pair. If the now see here, now here I changed this G and I replaced it with C and I got the same. Now this mutation, what is this type of mutation called? Same. Before the mutation also same. After the mutation also same. There is no huge impact. What type of mutation is that? What type of mutation is that? Silent mutation. Silent. Right? See here. I have put in the note. No change in the amino acid coded for. Why? Third letter is in the codon is a vowel. Right. If it changes, See here, if it changes A G G, I am replacing this G with U. So A U G, it is methionine, right? A G G, I am replacing G with A G U. I am getting a C R. A G G is A R G. See here, complete change in the amino acid. This changing is called as missense, which means one change in amino acid takes place. Change in amino acid change in amino acid can take place, right? Second point is it may or may not. These words are very important. May or may not impact the functional form of protein. Functional form of protein. What is the functional form of protein? Quaternary structure and tertiary structure. Do you agree with me or not? Functional form of protein, tertiary and quaternary structure of protein. You have studied this under unit number 2. Clear? Am I going fast? No, right? Can you understand this? Right. May or may not have an impact on the functional form of the protein. Right. Third point is it slightly, you see the words that are being used, slightly changes the primary structure of protein. Primary structure of protein. Primary structure of the protein means what they are trying to convey. Primary structure means amino acid sequence. Right? Is it clear? Now I have introduced you to three types of words. Right? Now if you want, just note it down. I said that when you come across the word gene, remember DNA segments. When you come across the word functional protein or functional form of protein, remember it is in its quaternary or tertiary form. Tertiary form. When you come across the word gene, functional protein, when you come across the word primary structure, right? Primary structure of protein, remember amino acid sequence. It is in its amino acid sequence form, right? If you want, note this down, right? You want to note it down, or shall I move on? Note it down. Yes. Okay. Right. So slightly changes the primary structure of the protein, right? Now, why this slightly change in primary structure? I have explained this on the last class. I drew a large number of diagrams and explained how a slight change take place, how a drastic change take place. Right? Even today, before starting the class, I gave that. I guess in the first page, I said that drastic changes are shown by nonsense mutation, insertion, deletion, chromosomal mutation. Minor change, slight changes are shown by silent mutation, missense mutation. Hope you can remember it. And last one here is it causes most of the time lethal, right? Lethal and deleterious or neutral effects are caused. Impact. Nonsense mutation, what does it? What happens here is after the point of mutation, After the point of mutation, if it come across a stop codon. Stop codon means UAG, UGA, UAA. If these three codons are come across, if they are made, then what happens is a nonsense mutation take place, which means they'll be causing premature termination of protein synthesis and they'll be stopping the coding. Right? Is it clear? Now this is the thing. Now see UAC. What is this amino acid? I mean this codon code for an amino acid. What is that amino acid? Use your codon book and let me know. UAC TYR. This is the actual one, original one. Now I am undergoing a mutation where I am replacing this C with a G. Now I get UAG. For UAG, what is the amino acid coded? Stop. Exactly. See here, after the mutation, if a normal codon is converted to a stop codon, then that type of mutation is called as nonsense mutation. Right? So after point of mutation, stop codon is formed. Right? What are the effects due to this? If you ask effects, first one is premature. Premature means before, right? Before it matures. Premature termination of protein synthesis, which means before the protein synthesis completes, it terminates, right? Premature termination of protein synthesis. The polypeptide's length has decreased. Polypeptide's length decreases. A shorter polypeptide will be produced. And last one, it is non-functional. The polypeptide that is produced with the less length is non-functional. Why? Normally, if you see the length actually it look like was, if you see now this is an amino acid sequence, and if this brings a stop codon, the length changes from 3 to 1. See here, compared to this has only 1. So it becomes non-functional due to stopping premature termination of protein synthesis. Before the protein synthesis take place, if it stops, it is said to be a non-functional one. Is it clear? Is it clear? Yes. Then that is the end of gene mutation, right? Now let's move to the insertion deletion part. So if you see insertion deletion for both of them, I'll give a single note. So insertion deletion can take place in one nuclear pair addition or deletion, two nuclear pair, multiple can be taken place, right? So what happens is if one or more nucleotide base pairs are added or removed, they are caused. They can cause insertion or deletion mutation. So what actually happens is when these take place, when one or more nuclear base pairs are inserted or deleted, they'll be causing a type of mutation. What is that type of mutation? Can you remember what type of mutation is that? If one or more nuclear pair is added or removed, they'll be causing the shifting of reading frame. If reading frame shifts, that type of mutation is called what type of mutation? Frame shift mutation, right? frame shift mutation. But there is another problem also. Now, normally, when one nuclear pair, two nuclear pair, four nuclear pair, five nuclear pair, when they're added, what happens is the reading frame. When if it is if this is the reading frame, when one is added, the reading frame shifts, right? So when the one is removed, the reading frame shifts again. So there's a shifting of reading frame take place. So just think now, if here if this is the reading frame, I actually read. When it shifts, the message I read will be changing, right? So insertion deletion. What is the effect that are caused by insertion deletion? Effects drastic change in genetic material, right? They can cause extensive missense. Extensive missense means what? Changing the amino acid sequence. Missense means changing the amino acid sequence. Extensive means an adjective, right? Extensive means, now just think, now here instead of choosing just missense, they are using extensive missense. Just remember it like that. Extensive type of adjective, right? These two are the effects, right? And right, there is another case that can take place, right? What happens is, for this insertion deletion, what if I write it here? All right, I'll write it like this, right? If triplets means multiples of three of nucleotide base pairs are added or removed, what happens? Now here, see, if one or more nuclear base pairs means 1, 2, 4, 5, 7, 8, 10, those numbers, not multiples of 3, right? I'll mention it here also, except for multiples of 3. Here, if multiples of 3 of nuclear base pair are added or removed, what happens? Is it hard to understand this? No, I'm just saying and going. I don't know whether you can get those. Is it clear? Or shall I say it slowly, explaining each of them? As it is a summary, I just went on saying them. Right? Now, if you have any doubts, I have uploaded the recording of the previous class. Right? So, do watch that. Right? I have given a complete explanation on that. Right? So, okay. Now, if three multiples of three nuclear base pairs are added or removed, what happens? The first thing that happens is reading frame reaches its original location, right? Reading frame will be now after shift. When if it is 3 or now if this is the sequence, now if we are adding triplets, the shift won't be taking place in this manner, it will be just adding it. Now just think, this is the reading frame sequence. I'm adding one nuclear pair, it will be shifting, the reading frame shifts. Now I'm adding a triplet, it will be getting back to its initial location. That is what they are trying to convey, right? Okay, reading frame will be reaching its original location. And finally, I mentioned all the important points. Reading frame will be reaching its original position, and uh, depending on importance on mutated regions, one amino acid can be added or one amino acid will be removed. Be removed. That's it. A small summary. Not small, right? It seems a bit big, but I don't think we can make it smaller than this. I have included all the points what are needed, right? I have included all the points what are needed uh, under gene mutation, right? So now see, now I didn't use a resource book here, right? To be honest. So this with the understanding, right? I'm giving this just to understand. Now, if now after studying the complete mutation part, right? You try to prepare a diagram like this, right? Don't try to memorize them. Just leisurely think that what comes to your mind under mutation. Just now think now mutation. Mutation is a two type. Okay, each mutation and each mutation what is classified. Why mutations are caused? What are the factors causing them? And how those mutations have an effect? What is the impact that is created? So those like a story, one after the other, you ask questions to yourself and you prepare it. Now here, when I am preparing, I am no sorting information by asking myself a question. Now, if you see insertion deletion, what is the impact that is created, how it is created, and those, right? Now, I'm giving here just for the purpose, for a easy access, right? If you want to quickly study it without taking whole resource books mutation part, right? You can use this for that purpose. I'm giving it, right? Okay. If you can prepare a summary which is shorter than this, that's really an achievement, right? Because you have prepared by yourself. Okay. Now chromosome mutation part, right? Ah, no, wait. There is another problem here. I have mentioned everything about gene mutation except one. That is uh important one about gene mutation is I mentioned here. I have space, right? Disorders. How many disorders are present for gene mutations? How many disorders? How many disorders? Students, can you remember? Two, right? What are they? First one, color blindness, and other one is sickle cell anemia, right? Now, I don't want to give the complete information about sickle cell anemia and color blindness. Let me give a small information, just a one information about that. So, with color blindness, if you see, it affects what chromosome? X chromosome that codes red and green light. Genes that are present in X chromosome. Wait. I haven't mentioned the genes part, right? Genes present in chromosome, green light, are affected, right? In sickle cell anemia, what happens? Sickle cell anemia, due to the mutated allele, what happens is glutamic acid in the primary structure of beta globin protein is replaced into valine amino acid. Valine acid. So when valine acid is caused, it causes abnormal folding. So this is the thing. Okay, perfect. Let's move to the chromosomal mutation part. Right? Chromosomal mutations are classified into two. What are they? First one, chromosomal mutation with respect to number. With respect to number. And the other one is with respect to structure. Let me first complete the structure part and then let's go to the number. Right? because that is how it is given in your resource book. So with respect to structure, there can be four taking place. Can you remember? Let's start recalling what happens. One is deletion, right? Other one is translocation. Eight. Duplication. And last one, inversion. Right? Deletion means loss of part of chromosome. Translocation means copied and moved. Duplication, moved to another chromosome, doubles the amount, or it increases the number of chromosome parts. Inversion changes the orientation of the chromosome. Right? Right. Wait, I'll separate this. Right? Okay. Right. Now let's see the effect. What type of effect are you might feel bit hard. You might say what does says is just keep on writing informations. It is hard for us. I feel that I can understand it is a bit hard because it is the first time, right? Summarizing all what we have learned, it a small structure. It is very difficult, and remembering that it is very difficult, right? Now, in behalf of you all, I am preparing a summary and I'm giving it because it will be useful for you all, right? Now, as soon as you prepare this, you might not, you know, it won't be sticking in your mind and you can't memorize it. It should be with practice, right? You should come across those. You should revise this 2 to 3 times, this complete part under mutation. I'm not saying memorize it. What you do is, there's a famous technique, right? Most of the students do is, there's a technique called 147 technique. Does anyone know about that 147 technique? Anyone of you know 147 technique? Students, feeling bored? Yes, feeling bored. Right. Just a few minutes, right? Within few minutes, we'll complete this part and I'll give a small break and then start the gene technology part. So for a few minutes, hold your energy up and be bit attentive, right? I asked, does anyone know this 147 technique is? What this 147 technique means? No. Any one of you know? No. Both of you. Now, this 147 technique means if you come across a new section, you have completed a new section of theory, and what you should do is not memorize, you revise, just read the complete section, allocating 1 hour or two. Now, for reading, for memorizing or studying, you might take this section. See this complete tute, right? You might take just think for reading. I'm just saying for reading, 1.5 hour, 1 hour, maximum 1.5 hour, right? So you allocate that time for the first day and completely read it. Just read it, understand and read it on the first day, right? At the end of first day, trust me, you won't be, you might not feel that you can remember all the information. That happens for every student. For the on the first day, reading every complete part from mutation, from the start of mutation until the end of mutation, which is the disorders part. After reading, you might feel that I'm lacking certain information. I can't remember at the end of the first day itself. Yes, that is the nature that happens, right? Don't lose that hope. What happens is after the first day, don't again revise on the second day. What happens is on the third day, take that mutation part again and again read it. Now see on first day what happens is first day you spent your time and you are reading that, right? On first day what was your goal? Reading plus understanding. Am I right? This was your goal, right? On third day you might have understood on the first day, but you can't remember. Understanding and remember are different, right? So on third day, you will be reading for the sake of memorizing or remembering. I guess the word remembering is better here, right? The second day, remembering. So on the third day, you will be get used to that content. While you are reading on the fifth day, what happens is sorry on not the third day, on fourth day, right? I have jumbled the days. Sorry. Right. Right, on fourth day, and here on seventh day. On fourth day, you will be reading for the sake of remembering. On the seventh day, what happens is when you are reading some portions, just you can just feel that the next portion will be on mutation, the next portion will be on aneuploidy, the next information will be on non-disjunction. Again, it will be disorders. You can predict on the seventh day. With practice, do this thing or this practice 147 technique for 1 week and let me know your results. Right, on the seventh day what happens is you will be reading and automatically it starts to keep on your mind, right? Because you have continuously read it for 3 days per week, right? And at the end, now here it will be memorizing automatically. You don't need to memorize him. At the end of this day, this week, trust me, now before this week, before it starts, just think you don't have 0% of this unit knowledge. I'm sure at least 75 to 80% you will remember. Trust me. Because this section is not a section we have been memorized. This mutation section. It is a content oriented theory part. Right? So just follow this technique for the next week. Or even if it is day. Yeah. Today it is Wednesday. Yeah. You can implement from next week or this week or Wednesday. And try it and let me know. I want the feedback. Right? Okay. Just 1.5 hours per day. Right? Okay. Right. We completed the effects part, right? Here. Effects. Deletion. Deletion cause detrimental effect. Just a word, detrimental. Translocation alters gene expression. Right? Duplication. Duplication cause uh, it also changes the gene expression. Right? For both, it can be used. Alters or changes gene expression. Inversion, adverse variations will be caused. Adverse. Right? These are the effects. And then we are coming to the chromosomal mutation with respect to the number. So with respect to number means we came across certain words, right? Let's recall what were those words. First word we came across was aneuploidy. What is this condition called? Aneuploidy means, now you are just bit familiar to these words. Aneuploidy means condition where one chromosome is more or less, right? And then we came across another word called non-disjunction. Yeah, before non-disjunction, aneuploidies take place due to what? causes. Why aneuploidy take place? Aneuploidy takes place due to errors in meiosis, right? Then we came across the second word called non-disjunction. In non-disjunction means in sorry inability to separate chromosomes during meiosis, right? Aneuploidy done, non-disjunction done. And then we studied about uh, trisomy monosomic condition, right? Let's include that also. What are they? Trisomy monosomic. Simply trisomic means 2n plus 1. Simply monosomic means 2n minus 1. Right? Which means one chromosome more, one chromosome less. Right? And last, we are coming chromosomal mutations and their disorders. Let's come to the disorder part. Disorders. Now, here, disorders, there are three disorders, right? What are they? Down syndrome, Turner syndrome, and Klinefelter. Right? Is it Tadan? Students? Let's continue after Tadan. Shall we start? Students, students, right, okay, okay. Right, last part, disorders. Right, here disorders, we know there are three. What are they? Turner syndrome, Down syndrome, and Klinefelter. So these three also can be divided, right? But ask how. How means we can divide it like this. First, no, I'll divide it like this. This is better. Wait, right. First, I can divide it like this and like this. This is aneuploidy in autosomal chromosome, which means one chromosome more, one chromosome less in autosomal chromosome. Other one is aneuploidy in which chromosome? Have a guess? What type of chromosome? First one was aneuploidy in autosomal chromosome. Other one is aneuploidy in. I want your answers. Aneuploidy in what type of chromosome? There are two type of sex chromosomes, right? Right. Right. When diploid in autosomal chromosome, we have only one disease or disorder. What it is? What is that? It is Down syndrome, right? Down syndrome. So Down syndrome is also called trisomy 21. It is a trisomic condition. Down syndrome is a trisomic condition in aneuploidy. So trisomic condition in autosomal chromosome, right? Right. Here we'll be dividing it into two. There are two disorders. What are they? First one is Turner syndrome, and the other one is Klinefelter. Klinefelter is simply we can say it as uh, trisomic condition, trisomy. At the same time, it has a genotype of X, X, Y. These individuals have a genotype of X, X, Y. Turner syndrome. Turner syndrome is monosomy, right? It has a genotype of XO, right? Hmm. So this XO, for XO condition, there is a type of word. What is that? They have given a term for XO genotype. Can you remember? Viable monosomy, right? I would have the space to mention it as viable monosomy. Uh, yeah, it is viable monosomy, right? So this condition is monosomy. This genotype XO means viable monosomy, right? That's it. This is our complete uh, summary on mutation part. See, right? Is it clear? This complete part. Clear. I don't know whether it is beneficial for all. I thought I felt like I want to give a small summary on this. So I hope this would be useful for all. Right? Right. So that is the end of mutation part. So what we'll do is now it is 12:26. Right. We'll have a 20-minute break. Right. A small break because we're doing a 4-hour class. Right. So 20-minute break. And let's meet at 12:46. Right. So be ready with your gene technology tube. We'll be starting gene technology. Right. Okay. Done. 12:46. Right. Right. Welcome back. Now we have completed mutations too, right? So we have completed mutations, and as we know, the final topic we are coming into is gene technology, right? Okay, let me get the tute. Right, this is the tute, gene technology. I hope I have shared this in your groups. Uh, okay, now before going on to this topic, let me give a small introduction. No, as usual, I am giving all. Uh, I'm giving a small type of introduction for each and every topic before starting. So similar to that, here also I'll give a small introduction. So we know that I said you on the first day itself, molecular biology, the subject or unit molecular biology contains subtopics, right? So DNA packaging, DNA replication, genes, operon, and chromosomal theory. Fourth topic was protein synthesis. Fifth was mutation, which we completed now. And last is gene technology. Right? Okay. Right. Now we know we have completed all these topics. Now we are going to start gene technology. Now gene technology also has certain subtopics. I said you that the complete lesson contain. Now we have completed almost 60 to 70% of a lesson. Right? Gene technology, it also contains some sort of content. Almost 30 to 40% of content is present in the complete lesson. So let's see what are the topics that are present. Gene technology, right? First topic where we'll be studying today, or we'll be introducing today, is DNA isolation. Second will be agarose gel electroporosis, and then we'll be introduced to DNA probe and hybridization, and then vectors cloning. Then comes DNA library, Then comes delivery system, DNA delivery system. Then comes DNA sequencing, DNA analysis, and then here restriction map. And finally, we have GMO, which means genetically modified organisms. Right. Okay, sorry. Yeah. Right. Now this is what gene technology, the final tute also contains. Now if we complete all these topics, which means we have completed unit 7, right? So don't worry, there are many topics you might see. So there are multiple topics, right? If you see compared to these topics, you might feel so there is large number of topics here, right? But there are a large number of topics, but each of them are small content, right? If you see DNA isolation, aggressive electrophoresis, inshallah, we can complete this by today. Right, next class we can complete these sections are smaller. Here in this complete part, GMO is the part which has some sort of huge content, right? Okay, then that's it, we can go into the lesson directly, right? Okay, right. Gene technology. Tools, techniques, and methods in gene technology. This section examines the process of gene technology starting from DNA isolation through the identification of desired DNA sequence to gene technology to recombinant DNA technology. Now, here what we'll be learning is now up until now what we have learned. Just shortly, we will revise. Under DNA packaging, we learned how DNA is arranged into the nucleus of prokaryotes and nucleus of eukaryotes. In DNA replication, we understood how a master copy of DNA can be used to produce multiple copies of DNA. In the third, genes operos, we understood how genes are present, how DNA is scattered, and how the chromosomes are present. We understood that. In protein synthesis, we understood how from DNA proteins are synthesized. DNA was converted or transcribed into an mRNA. The mRNA's information was converted to amino acid sequence, and this was converted again to further polypeptides. After modifications, the polypeptides converted to proteins. And under mutation, we learned what if an error take place. So those errors can lead to mutation. And what are those mutation? Gene mutation, chromosome mutation. What are those ages? I gave a small idea or summary, right? Now here what we learn is how can we use this knowledge in applications. That's what we are going to learn. Now if you see now if you take a DNA, can we insert this in DNA into another organism? Yes, we can. So how? That is one question. And how we can take DNA. We'll be learning those. So we'll be learning how this knowledge on molecular biology can be applied on various applications. Right? That is what we are going to learn here. Right? Right. That is what they're trying to convey in the first part. The isolated DNA needs to be cut into different pieces of DNA must be joined and times it should be copied in vitro. And now, before going on this section, there are certain words you should understand: in vivo and in vitro. In vivo means inside the living organism, right? In vitro means outside the living organism, right? In vivo means inside the living organism, means inside of a human body. Just think about like that. In vitro means outside the living organism. Example, laboratory conditions, right? Now, the first step in gene technology was, now I give a diagram, right, is DNA isolation. DNA isolation over here, before starting, I'll give a small idea what happens here. Now we know that DNA is present in the nucleus of eukaryotes, right? Think this is the nucleus and this is the cell, right? Now what we should do, we should extract this DNA out in order to insert this DNA into another organism. Am I right or wrong? First, to insert a DNA, we should isolate the DNA. Now, DNA is present in nucleus, right? Am I right or wrong? Hmm. Yes. Okay. So that is what we'll be learning here, right? So here there are certain steps how these can be done, and we'll be studying those under DNA isolation. But here they're giving a just smaller idea. The isolated DNA needs to be cut. They should be cut, it seems. Uh, and then they're saying they should be joined and they should be copied. See here, they have said three main important activities that should be done. What are they? They should be cut, they should be joined, and they should be copied. These three are the instances which are involved in DNA isolation, and at the same time, see here, what are they? Cut, join, and copy. For these three things to take place during DNA isolation, we need some sort of compounds called enzymes. Is this the first time you're coming across this word, enzymes? No, right? You have come across these enzymes. Now we'll be understanding what are those enzymes, right? These involve several enzymes. See, these involve several enzymes acting on DNA. Distinguishing a unique DNA sequence from the rest requires separation of DNA based on the size of fragments. To detect a specific sequence of DNA, we should separate them based on their size, it seems. Right? In making genetically modified organisms, this DNA needs to be transferred to a recipient organism using suitable method. This isolated DNA should be transferred to an organism. If those DNA are transferred to an organism, that organism is called what type of organism? What type of organism? Genetically modified organism, right? Copying of DNA is possible in vivo. In vivo means within the organism, right? Using cloning, it seems. If we need to copy DNA within the living organism, we can perform cloning. And if vitro, in vitro means we can perform polymerase chain reaction, PCR. PCR. Have you come across this word? Not in your syllabus, but in your life. I guess you might have come across this word, PCR. When? When have you come across this? I guess 2 years ago, 2 to 3 years ago, during the COVID-19, right? Now we'll be learning about that also. PCR. What is PCR? How PCR is used? How it takes place? What is the purpose of PCR? All we'll be learning here, right? Okay, right? Is this clear? This first part. See here. Now we know enzymes are required. Why enzymes are required? Enzymes are required to cut the DNA. Enzymes are required to join the DNA. Enzymes are required to copy the DNA. Right? Just go through this first part, the introduction part, and let me know are there any doubts. Clear. This is clear for all. Clear, right? I hope it's clear. Right. Now we are coming to the topic, which is isolation of DNA. Sorry. Before going directly into the topic, which is DNA isolation, they are giving some sort of information. That is how they are introducing all the lessons. It is not new to you all. Before going into direct information, they're just giving information, just giving a small warm up session. They're giving some sort of guidance. What is this? How it is happening? Those stuff. Why it is used? Gene technology begins with the isolation of target DNA. Correct, right? Gene technology means the technology or the application of DNA. So they are saying it begins with the isolation of DNA. Correct, right? Before inserting a DNA, what should we do? We should isolate it from the cell, right? DNA sequence from the total genome of the donor cells. Genome means, I hope you can remember what is genome. What is genome? If you can't remember, genome means collection of gene, right? Exactly, collection of genes, right? Very good, very good. Right, right. Okay. Purified DNA. Purified DNA means extracted DNA is required for many applications. Now this DNA can be used in various applications, it seems. They are given. It can be used for studying the DNA structure and chemistry, right? Examines DNA protein interaction. See here, studying DNA structure and chemistry. Uh, you might you might think, sir, this might be how James Watson, Francis Crick said that DNA is a double helical structure. Correct. Do you agree with me? Now, using the application of DNA isolation, DNA isolation can be used in studying the DNA structure and chemistry. It seems it might be using this. James Watson and Francis Crick said that DNA is a double-stranded molecule. It is used for examining DNA protein interaction. DNA protein interaction means during DNA packaging, DNA and histone proteins interact, right? So these can be studied. Carrying out DNA hybridization. Now these will be understood in today's. This will be understood under gene technology institute, right? Carrying out DNA hybridization. You might ask, what is hybridization? Just think pairing, right? Okay, this is one DNA strand. This is another DNA strand. These will be paired, right? If these are paired, this is called hybrid idea. Today, not becoming hopeful, right? Sequencing, sequencing, PCR, and all of these will be coming across in the next class, right? Now, for the time being, just try to understand these are the applications where we can introduce DNA, or these are the industries, or here these how DNA can be applied. DNA's isolation can be used application of studying DNA structure. It can be used to examine DNA protein interaction and carry out DNA hybridization, sequencing, PCR, performing various genetic studies. Since DNA molecules are very long, the entire length of DNA molecule cannot be isolated except for shorter DNA molecules. Such are right. DNA molecule is a long molecule. So if we want to insert this into another molecule, we can't extract the complete DNA molecule, right? Instead of that, what we'll do, we'll be cutting them and we'll be extracting. Can you understand how Enzymes are used in this process. Enzymes are the ones that are useful in cutting the DNA molecule and extracting, right? However, breaking the DNA or shearing has to be minimized, right? Now, this is the thing. Now, you think, now here I said that this is a DNA. Normally, DNAs will be looking like this strands, right? See like this. Now, we can't extract the complete sequence. So, what we'll do is just think this is the portion we require. We'll be taking an enzyme. Now, I'll be introducing that enzymes. We'll be learning today. That enzyme will be cutting this sequence and it will be separating this sequence only. And it will be inserted into an organism cell, right? But they're saying even though we follow this, right? Now here enzymes are used, but breaking the DNA and shearing should be minimized itself, right? Breaking the DNA. Now enzymes, what they'll do is they'll be introducing breaks enzymes. But if humans start to break them, what we'll do is they'll be causing various ill effects. That is what they are trying to convey. Breaking the DNA and shearing should be minimized due to extraction process because it can now see because DNA is a sensitive component. Now the complete generation is dependent on this DNA, right? While we just cut them, if there is any sequence that is being removed, any essential information, a complete loss, right? That is what we learned under chromosomal mutation. Why? During the deletion part, I said that cutting of a part of chromosome contains genes, which means loss of a gene is a deleterious impact, right? Similarly, here also cutting. Now, if you use enzymes, what they do is they'll cutting. That is why they say enzymes have specificity in their function. Can you remember? Under unit 2, we understood that their specificity. Enzymes are specific, highly specific for their function. Can you remember that point? In unit 2, under characteristic features of enzymes, we understood that enzymes are highly specific in their reaction. That is why we will be using enzymes here. So instead of we humans contributing them, we use enzymes for that. Right? Right. Just read this part and let me know if there are any doubts. This isolated part, DNA. Clear. Okay. Okay. Perfect. Right. This part is completely clear, right? Then let's move to the next part. Right. The basic principles and major steps of DNA isolation are these are the steps how DNA is isolated. What are the steps? First one, homogenization or disruption of cells. Second one, inhibition of DNAs. Third one, dissociation of nuclear protein complex, removal of contaminating materials, precipitation of DNA. Right? This time, 2025 A level, they have asked this question. Trust me, they asked this as an essay, right? This can be asked as essay. So I'm not trying time to tell that this will be asked again in the question in your batch 2026, but they can ask this in a structure, right? right? So, uh, not hard part, it's a very, very easy part, right? So, these are the steps. DNA isolation can be done in these steps. First one, homogenization. Now, I explain what are these steps, right? First one, homogenization or disruption of cells. DNA is located in the nucleus of eukaryotes and it is concentrated in the nucleoid in prokaryotes. No doubt about that, right? We have learned about this in DNA packaging too. The first step in DNA isolation is releasing the DNA from the cells by breaking the cells, right? So to isolate a DNA, it should be broken or lysis of the cell. Lysis also has the same meaning similar to breaking the cells, right? Cells can be lysed, lysed means broken by mechanical methods such as grinding the cells, homogenization, or enzymatically by lysozyme, right? Now see, homogenization disruption of cells. First step, what actually says is for this DNA to be isolated, it should be extracted from the nucleus first. It should be isolated from the nucleus. So it can be done using two methods. First one is mechanically, other one is enzymatically. If you perform mechanically, we can do it by grinding the cells. Or second one, we can do it by homogenization. Oh, you can ask what is this homogenization? It is also type of mechanically removing the DNA. They haven't given much information about that. Just remember, homogenization means the homo means what? Similar type, right? So similar type of DNA will be extracted. It is a method like that. Enzymatically, we can introduce an enzyme called lysozyme, and we can remove. Can you remember lysozyme? Is this is the first time you're coming across? Is this the first time you're coming across lysozyme? No. Where? Where have you come across this lysozyme? Immunity. In immunity, we came across this lysozyme and external barriers. They are present in the saliva, tears. These are present. So what they will do is, if this is inserted into a prokaryotic cell wall is present, right? Lysozyme enzymes will be breaking this cell wall. After breaking the cell wall, it can be easily removed. The DNA can be easily removed, right? So what are the ways? It can be done using mechanically or it can be done enzymatically. Mechanically, it is done, it can be done in two main methods: grinding and homogenization. Enzymatically, it can be done by introducing lysozyme. Lysozyme is an enzyme which can break bacterial cell walls. Is the first point clear? For the DNA to be isolated, we should break it first from the nucleus, right? Now think like this. Now, if a person is to be taken out to a other country, first he should be extracted or he should be taken out from his home, right? Before he goes into various other countries, before he goes to foreign countries, first he should be taken out of his own home. So similarly, for the first step is taking out from his home. So DNA is extracted from the nucleus or from the nucleus of prokaryotes by this method, the first method, homogenization or disruption of cell. Is that point clear? The first part. Clear, right? Okay, perfect. Second one, inhibition of DNAs. Now, this is the thing. Now, just think. Now I can just go on telling the theory part. Just to make it more entertaining and a bit interesting, I'll use certain examples. Now, just think. Now, just think there's an individual, right? He has been under the guidance of his parents until an age of 15. He was completely present in his home, right? Uh, he'll be going to his school and coming, but he does not have any friends associated, right? He's kept in his complete home surrounding. So just think after 15, 16, he gets a student. No, he's a now student. Right? After 15, 16, just think 18 something, he comes to his advanced levels. Now he has to go on to classes, right? So what happens? He has to come into the external world. So until that, he was in the house and he was just maintaining. He's spending most of his time in home. and he's not more exposed to the environment, which means socially he's been completely neglected. He's been just now negra, right? So he's been completely under his supervision of his parents. Now just think, after 18, now he's coming to the external world. Now he's coming across, now he's coming to the external world for going classes, for various other activities. Now just think, there is a group of people in the environment or the towns that normally they are present, in the society they are present, where there are bad influences, right? There are some sort of people called bad influences. Now just think, they can be some drug addicts, they can be womanizers, they can be various politicians, right? They can be any of them, but they are given a common name which is called as a bad influences, right? Now just think, a person who is coming out of the house, just now he's coming at his age of 18, he's coming to the external world of the society, just to get become a bit more used to the environment, and he wants to go to classes and various activities. Now he has to come out. Now just think, these bad influences, will they target this person, or will they target just random person, which is easy for them, targeting a person who's coming for the first time, or for a person who is already present in the society actively? The new person, right? Why? Because he does not have any sort of knowledge about the external world. For him, his parents and his siblings. Now he was a child who was under supervision of his parents, and he knew, he accepted all what his parents said him. So he's coming to the external world, for the external society. Now these people will be targeting him. And they'll be trying to mind wash, giving them brainwashing some sort of information. They'll say, now you have a great future in this side, you go to this side, and they'll be starting to use him in various purposes. Right? Because he does not have any sort of knowledge of current situation of the external world. Right? Similarly, what happens here is, now this extracted DNA is similar to some sort of this individual. Why? DNAs are normally present in the nucleus, but we need the DNA to be extracted now during the DNA isolation. Now, as the DNA is extracted out, these bad influences here, some sort of bad influence called DNAs comes, right? These DNAs, what they'll do is, they'll be degrading the DNA. When the cells are broken, the DNA may get come in touch with the DNA degrading enzymes such as deoxyribonuclease, right? Therefore, DNA must be protected from such enzymes causing shearing. So what they do is, now these bad influences will be coming in. What they'll do, they'll be starting to brainwash. They'll be giving sometimes they'll be threatening him. They'll be starting to give him bad drugs, and they can make him fall into that trap. Right. Similarly, when the DNA is extracted out, these DNA's enzymes will be coming, and it will be coming as a friend. Now here also, these bad influences, they'll be coming like a friend, like they'll be coming and they'll be speaking, you have come to the okay, you're coming here to classes, okay, right. And they'll be first, for one day they'll be talking, second day also they'll be okay, okay, today was a class, okay. Then they'll be frequently coming, and they'll be free after meeting frequently. What they'll do, they'll be starting to share their feelings, they'll be starting to share their informations. After sharing, what happens, they'll be starting to maximize their opportunity of that relationship. Similarly, what they'll do is, DNA also comes as a friend, but what it does is, it will be degrading the enzyme. Degrading means it will be altering its properties and information. Is it clear? Right. Okay, clear, right? For this to be prevented, they are saying chelating agents are added to remove metal ions required to nucleus activity. Chelating agents. Right? Now here chelating agents means similar to like now he has no friends, right? Because he has no friends, he goes in search of a friendship. He goes in search of a shoulder where he can lie and give some, share some feelings. That is why he's being exposed to this bad influences, right? So in place, if he has a good friend, will this good friend leave him to be under influenced by these bad influences? Will this good friend leave him? No, right? He won't be letting him to go to that bad influences trap. So similarly, here to prevent this degrading activity, we can add some sort of agent called chelating agent. What the chelating agent does? It removes the metal ions required to nucleus activity. Now, this statement they haven't given an explanation in your syllabus, right? They haven't given an explanation for this. They haven't given an explanation for this statement in your syllabus. I'll give a small explanation, right? You don't write now what I'm giving now an explanation. Don't write these information in your paper. If they ask, don't write these because just I'm giving for explanation. Now see, now if you just read, it removes the metal ions required for the nucleus activity. Can you understand anything? Just be frank and let me know. Can you understand that? We can understand that chelating agents protect them, but how? Can you understand how? No, right? Can you understand? No. So, for your understanding purpose, I'll explain what actually happens, right? Right. Now this is the thing. This is DNAs, right? Now, you know cations, right? Cations, magnesium, sodium. Cations means ions or elements that are in the positive ionic form, right? Sodium Na plus, Na plus, magnesium 2 plus, cationic form, right? Now I'm taking magnesium, for example, right? I'll take magnesium. They are saying metal ions, right? Now magnesium is a metal. I hope you have studied this under your chemistry, inorganic chemistry. Magnesium is a metal, right? Think magnesium. Now, what magnesium means? Magnesium is a type of substrate that comes and binds to this DNAs. Is it clear? Now, what happens now? See, now it will be looking like this, right? Magnesium, right? Now, tell me the complete complex. What is the charge of the complete complex? Now, DNA had an initial charge. DNAs had an initial charge of 0. It had a plus 2. Now, the complete complex. This complex, what is the charge of the complete complex? Let me know in the chat. What is the complete charge? Plus 2. Exactly. Plus 2. Right? You know that opposite poles attract. So here plus charge is present. It will be attracting to a what type of charge? Negative charge, right? So what happens here is we know that DNA. DNA are charged or non-charged molecules. DNA is DNA a charged molecule or non-charged molecule? Just let me know from the idea. I don't want the correct answer. What you think is DNA charged molecule, non-charged? You are saying non-charged, okay? I don't want the uh, assumption from both of the students. What is your point of view here? Is DNA charged molecule or uncharged molecule? Non-charged. Both of you are saying non-charged. Let's see. Now, this DNA, it looks like this. Will you agree with me or not? I'm speaking about this. Now, I'm taking this solo portion, right? I'm taking this portion. This portion looks like this. You agreeing with me or not? Wait, wait, wait. I'm speaking about this portion. One nucleotide. You are agreeing with me or not? What is this? This is a phosphate group, right? What is this? A pentose sugar. If it is DNA, what is the type of sugar? Deoxyribose. Now, this can be nitrogenous base. It can be adenine, guanine, cytosine, thymine, anything. Now, phosphate. This is the problem. Phosphate, how does it look like? P, double bond O, double bond O, O, O. Here minus, here minus. PO4 3 minus. So here we know phosphorus has a valency of 5. So one double bond should be removed. So actually it will be looking like this. So see 1, 2, 3, 4, 5. Right here a 5. So this one is completed. So this it looks like PO3 minus. Do you agree with me or not? Agreeing, right? Okay. Now we know that these phosphates are facing the outside of the DNA molecule. The phosphate and the DNA, the pentose sugar present, so they'll be forming a sugar phosphate backbone in this manner. So the phosphates are faced in this manner outwards. Clear? Is that clear? Phosphates are faced outwards. Yes. Now tell me, is DNA a charged molecule or uncharged molecule? If you see pentose, no charge. Nitrogenous base, no charge. Phosphate, does it have a charge? Yes, phosphate has a charge. What type of charge? Minus 3. So, phosphate will be forming, it is an anion. So, due to the presence of this phosphate, only due to this presence, DNA is a charged molecule. Charged molecule. Right? Now, think about this now. The complete complex has a charge of plus 2. It is attracted towards this DNA molecule, which has a charge of minus 3. Right? So, will you agree why DNAs comes now? Can you understand why DNA is coming like a friend to attract with the DNA molecule? Can you see why it is coming? It wants to neutralize its charge. Now I am explaining you with respect to one magnesium, one DNA molecule. You might see, sir, might ask me, sir, now this has plus 2, this has minus 1. So the complete charge will be getting a minus 1, right? You might ask like that. Now, this is the thing. Now, is only one active site is present for enzyme molecule? No, right? There are multiple, right? Think like this. Sorry, sorry. Right. Here one magnesium, here one magnesium, here one magnesium. So total charge, what is the charge look like? Plus 2, plus 2, plus 2. So how many DNA molecules are required for degrade or the neutralization? How many DNA molecules? So the complete complex has a plus 6 charge. Now this is a DNase enzyme. Don't confuse that. DNase enzyme. How many DNA molecules are required uh, to neutralize it? Exactly 2. Right? Why? One DNA molecule is minus 3. So if it comes like this and binds and comes somewhere here and binds, the complete complex, this complete complex becomes zero charged. Now, due to this binding, DNase is benefited. Why? Its charge is neutralized. But the problem is DNA will be degrading its enzyme, degrading its activity. Now, DNA has a double helical nice structure like this. The phosphate backbones are located nicely. Due to this attracting, it will be pulling, and sometimes it will be looking like this, and the shape will be lost. Right? It's specifically its information will be affected. Everything will it is completely degraded. Its nature is altered. Can you understand what why DNA is being affected by this? Can you understand how DNA is affected by this binding? Yes, DNA is to complete or fulfill its purpose. It is choosing the DNA, right? But DNA normally it is benefit. No, if the DNA remains normally like this, it does not worry or bother about its negative charge. It is happy what he's got off, right? He is thinking that I have got minus 3. I'm happy with that. Alhamdulillah, he's bit happy and satisfied. But DNA is what it wants is it has plus 2 charge, but still it wants to neutralize that plus 2 with gaining some charges, right? It is not satisfied. Now this is a comparison between a satisfied person and not satisfied person, right? Okay. Now what we'll do is to remove this. Now all this happens because of what? This iron, right? Initially DNA was non-charged. Just because this magnesium came in, it became a charged molecule. All because of this charge only DNA is tending to attract. So what if we remove this magnesium ion? What if we remove the magnesium ion? Will DNA tend to react with DNA? Can you understand my question? No. Ah. To remove this magnesium only, we are adding some sort of agent called chelating agent. So chelating agent, what it does, it will be removing the magnesium ion. So DNA has nothing to bind with. So it will be non-charged. So it can't react with DNA. Now see this statement. Can you understand the third statement? Clear, right? Right. So is this second point clear? Inhibition of DNAs. The complete second point, is it clear? As soon as we expose the DNA, the second step is we should protect that DNA. Because we remove the DNA, we are not achieved what we want. We have to protect what we extracted. So the second step is mainly based on how we can protect it. Clear, right? Shall I move to the third point? Okay, perfect. So third point says that dissociation of nuclear protein complex. I guess you might have some sort of knowledge. Now, normally DNA and histone proteins are looking like some sort of uh, very good friends because they are every time associated to produce a chromosome. But now, as they are very good friends, can we take everywhere both of them? Now, just because you and your friends are best friends, can both of you go to everywhere you are going? Can't, right? Now you just think. Now both of you are very good friends, right? Now there is an opportunity where your one of your friend is taking a level science stream. Now other one, just because he's your best friend or she is your best friend, Does she also can go to science? No, it depends on her choice, right? So just because they are very good friends, we can't take everywhere those. Both of those friends cannot be taken everywhere. So similarly here also DNA and histones are very very close related. But still here during DNA isolation, see here DNA isolation. During DNA isolation, what we are focused to extracting what? During DNA isolation, what is our main goal? During DNA isolation, what is our main role or main goal? The word still says DNA isolation. What is our goal? Students, isolating the DNA. Simply that is the answer, right? Isolating the DNA. So, do we need to consider about the proteins? Do we need the proteins actually? We want only the DNA. We don't need the protein. Yeah, exactly. So, what we'll do, we have to remove that DNA protein interaction as well. Just because they're very good friends, we can't take those both also. We have to extract the DNA and insert only the DNA, right? So this is the third step. This is focused mainly on extracting the DNA only, protecting it from proteins or extracting it separately, right? DNA needs to be freed from the proteins that bind to DNA. Indirectly, they are saying which protein? Histone protein. DNA protein interactions are disrupted by sodium dorsal sulfate, phenol, proteolytic enzyme, right? These three are the agents which can be added to remove this protein and DNA interaction. So SDS means just put a bracket and write sodium. I'll write here sodium dodecyl sulfate, right? Right, by adding these three we can uh, dissociate the nuclear protein complex. Is that third point clear? Right. Clear, right? Perfect. Now we are coming to the fourth. To understand the fourth, you should have some sort of knowledge. See, now from the cell, we are extracting the DNA. Yet you are agreeing with that. Now, do you think the DNA is extracted like a crystal? Now, DNAs are normally nanometers in size. They are very, very, very small. Now, see, the nucleus, the nuclear envelope size, can you remember? Which we learned under unit 2, 20 to 40 nanometer, right? So nanometer, if the nuclear envelope gap is 20 to 40 nanometer, what about the DNA that is present in the nucleus? Very, very, very small, right? So do you think this can be removed as a crystals during DNA isolation? Do you expect we are extracting them in the crystal form? What about that? What's your idea? Students, just your idea. What is your thought on that? Are DNA isolated as crystals or like this shaped DNA molecule? Do you think it will be coming like that? No. These will be taken in aqueous form. Aqueous form, which means in liquid form. The DNA will be contained in that liquid, right? That is how we take. Is it clear? Is it it is a bit confusing, no, right? Aqueous form is simply liquid form. That liquid will be contained. If you see, it will be aqueous form. This aqueous contains DNA, but it cannot be seen by a naked eye. Is it clear? Right. Okay. Now, in the steps, we are coming to the fourth one. Or fourth one is removal of contaminating materials. Even though we remove the proteins, there are certain other contaminants too. Contaminants means simply if we see the aqueous solution, except for the DNA, all other molecules are considered to be contaminants. Will you agree with that statement? Except for DNA, all other materials that are present in the aqueous form are considered to be contaminants. You, do you agree with that? Yes. Okay. Then you can understand this. All other molecules in the cell are contaminants of the DNA. Removal of these contaminants is required for some other applications, right? For some applications. So extracted the DNA are used in various applications. That is correct, right? Right. Now, all after remove the contamination also. Now, what is remaining after remove the contamination? It contains the aqueous form. In the aqueous form, DNA is present. To this form, after the fourth step, does it contain any contaminants? After the fourth step, no. What the final aqueous form look like? What it contains? By the time it comes to this fifth step, the aqueous form, what it contains? DNA only. Very, very good, right? You want that idea. Now, DNA is present. But here is the actual problem coming. Now, think like this. Now, we have to insert why we are performing this to isolate the DNA only. Now, if we want, why are we isolating the DNA? We should understand what is the main purpose of that. Now, actually, why we are isolating the DNA is we have to insert this into another organism. Do you agree with that or not? You agree with that, right? Okay. But can just think practically, will it be practical if we insert the DNA in aqueous form? Just pouring it as a liquid. Is it practical? Can we pour the liquid into a cell's nucleus? Is it practical? No, right? Here we need the DNA as a form. It should have a solid-like form. For that only we are doing this purpose. Precipitation of DNA. Precipitation means normally what does a precipitate mean? Now, if you see, if this is a solution, just think adding something gives a suspension like this. Suspension, solid like suspension. This is called as a precipitate. PPT. Normal chemistry students, you might use these as PPT, precipitates, right? Now we have to precipitate DNA. We should precipitate as a solid suspension. Can you understand what is our idea here? Why we perform this fifth step? Can you understand why we perform this fifth step? Yes. Okay. Here the DNA dissolved in the aqueous phase is precipitated with cold 0 degrees ethanol. What we do is this aqueous solution. Now what we do is we'll be reacting it with 0 degrees ethanol. 0 degrees ethanol means it looks like a gas-like compound. And after precipitating, we'll be reacting with 0 degrees ethanol. What happens then? The precipitate is usually redissolved in buffer. Uh, After dissolving it or reacting it with 0 degrees ethanol, it will be giving a precipitate. What does this precipitate contain? DNA only, right? DNA, right? Now, what we do is we will be taking the precipitate and they are saying they'll be re-dissolved in a buffer. What they'll be doing? What they'll be done? They'll be taking the precipitate of DNA and they will be re-dissolved in a buffer. Now you might ask, sir, what is this buffer? Buffer means just simply remember it as a regulator, regulating solution, right? Now here, just think the DNA we extracted. It might contain. Now we have what we have done. We have taken it the DNA. We have taken the DNA. We have removed the the protein complexes, we have removed the contaminants, we have taken it and we have dissolved in 0 degrees buffer, so 0 degrees ethanol, and we have dissolved in buffer. Now, until we dissolve it in ethanol, right? After dissolving in ethanol, do you agree? Now, can you tell me? Can you answer this question? Won't there be any pH changes? Won't there be any pH changes? Why? Because we are dissolving it in various agents. See, first what we do, we are reacting with an enzyme, and then we are reacting with chelating agents, and then we are reacting with sodium disulfide, phenol, or proteolytic enzyme, one of them. And what we do, we'll be again removing the contaminants, and then again we'll be dissolving in buffer. Won't there be any pH changes? This much of dissolution, this much of reaction. The initial DNA might contain a pH, right? Will there be a pH change or not? Well, there might be a change, right? So there will be a change or there might be a change. Yeah, both of them are correct, right? So inserting them. Now, think actually in a cell, this is how DNA looked like. Now, this contains some sort of conditions. It contains some sort of length. It contains some sort of information. It contains some sort of pH. Now, at the end of the process, DNA isolation also, if we get the same DNA with the same length and same info and same pH, that is a beneficial process. Why? This is what we expected. This is the DNA what we expected, and that is the DNA which have we obtained. But due to this dissolution, there can be pH changes that take place. If there is a pH change that has taken place, is this process efficient or not? Not efficient. Simply, now your fathers might have done this. The businesses, vehicle businesses, there are various sites that are present for vehicle businesses. Rear 7, ICMAD, right? So when we see the picture there, the vehicle picture, might have posted the best pictures of the vehicle, right? While after buying that, now we are seeing we are comparing that. Now, if we find there are certain changes, now will we consider this to be an efficient business? No, right now. See, initially it was looking like a vehicle which had perfect. There wasn't any there wasn't any actually it was a perfect vehicle, right? It was a perfect vehicle. The wheel, alloy wheel, mileage, everything was perfect. Now, after we see it and after we are gonna purchase it, now if you are seeing there are certain changes. The headlight has gone. There's a bump, and we are seeing there are various changes. Is it an efficient business? If we purchase it, I'm asking if we purchase it, is it an efficient process? Yes or no? Photo or ghati where nearly where mari. No, right? Right. Exactly. So here also the same thing. Why? Because now initially what we expected, we wanted this DNA. At the end also we want that DNA. We don't want any changes. To prevent that changes, what we do, we'll be dissolving it in a buffer. Buffer, what it'll do is it will be regulating the pH. It will maintain that pH and prevents it from changes in pH. Is it clear? So simply buffer means just think for this under this portion it is a pH regulator. They won't be asked what is the function of buffer and everything. Here it is acting as a pH regulator. Right? Last point. RNA is removed by limited treatment with DNase free RNAs. Right? If there are any RNA that is present, right? There can be RNA, right? If there are any RNA present, it can be removed by limited treatment with DNase free RNAs. What is that meant by? Now you would ask, won't this RNAs react with DNA molecule? No, they are specific, right? They are specific. They'll RNAs means ribonuclease, right? Nuclease means degrading, like so. RNAs will be reacting only with RNA. DNAs will be reacting with DNA, right? So just think now, like this is the thing. You might ask, sir, now what we did, we was extracting the DNA, right? How does an RNA come at the middle? Do you have that question? Do you have that question? Right. Now this is the thing. Think like this. Now if you find like this, and if there is an RNA molecule, now here there can be RNA fragments, right? Now see, this can be an RNA fragment. But you might ask, see, gee, this belongs to adenine also, right? Yeah, it can belong. But now we are seeing as a sequence, right? Actually, there can be RNAs present, right? Now we'll now see that X the solution during the removal of contaminants. Now see here, I guess this will be see. Now during the first process, what we did, we were removing the DNA from the actual location, from the nucleus or nucleoid, right? Second, what we did, we removed or protected the DNA that is present by reacting it with the metal, reacting with chelating agents. But see, in these four steps, they haven't mentioned how to remove RNA. Can you see? By undergoing these four steps, we can remove all the components except for RNA. Why? RNA is uh, RNA is similar to uh, a traitor. Now, you know the word traitor, not an enemy. You know the word traitor. Do you know the meaning of traitor? No. Yes. Nambikadrohi. Right? So defeating an enemy is easy, but traitor is difficult. Why? Why? Because he knows all your strengths and weaknesses. Am I right or wrong? Enemy, he tries to search, but traitor, he knows this is your strength and this is your weakness. So defeating a person who knows your strength and weakness is very difficult. Is it correct or wrong? Correct, right? Okay. Now, if he knows, if the traitor knows your all features, your strengths and weaknesses, he is similar to you. Why? You two are the ones who knows your actual weaknesses and your strengths, right? So similarly, if you see here, RNA is similar to a traitor. Why? RNA is also containing the same features of DNA except for the ribose sugar, single stranded, and the uracil base. Or except for these three features, they are all one the same. Can you understand? Can you understand? Yes. So some sort of a person now, a person or a molecule like RNA, which has the same features like DNA, cannot be removed by these simple steps. It cannot be removed by these four simple steps. For that, we should create a master plan. To defeat a traitor-like person, we should have a master plan. That master plan is this. So all after doing this, we'll be adding a DNAs free RNAs. That will be removing the RNA. Is that clear? RNA ഇവരോടേ molecule മാറി. Both of them have the same functions too. Why? They are involved in genetic material. They have different functions at the same time, but they have the same features except for a few differences. They are somewhat similar. So to remove some sort of molecule like that, what should we do? Should have a master plan. We can't remove it initially. What we'll do is we'll be bringing them closely until the last point, and at the end, before the process ends, we'll be removing it. That's the idea. Is it clear? Clear, right? Perfect. Now just go through this part completely and let me know if there are any doubts. If no doubts, then If there are no doubts, then perfect. Just go through it. Sir, is DNase free RNase the whole name of the enzyme? No, no, no. DNase free RNase. RNase is the name. DNase free means it does not contain any contamination of DNase. Why? If DNase is present, it can remove DNA, no? Right? Now, if even if DNase is present, it will be removed in the second step. Can you understand? Why? We are adding chelating agents, right? Chelating agents will be removing DNase, will be adding DNase. But if RNAs are present, RNAs will be removed in this. RNAs will, if RNAs are present, RNAs will be present at the end, and these RNAs are the ones that are removing RNA. Is it clear? Yes, clear, right? If DNase is present, just think if DNase is present here, it will be removing the DNA as well. So then it is a completely useless process. Why? The main motive is to retrieve or gain the DNA or extract the DNA. If you remove it at the end without any purpose, that does not have a efficiency, right? So initially, if you see here, if there are any RNAs, see here if in the cell, if there are any RNAs, it seems like a bit of story, right? It is like a crime thriller movie. If there are any RNAs present that are initially in a cell, in this checkpoint, normally degrading enzymes are removed. Inhibition of DNAs this time. But here, chelating agents will be only focused on DNAs and it will be allowing RNAs to move on. You go. It will be allowing to move on. Now, at this point, the cell may look why. No, they will be looking why RNAs is being permitted to move. But at final, it will be giving a twist and it will be removing the RNA if it is present. You understand? Right. This is a type of master plan. Is it clear? The complete part? Just let me know if it's clear. Clear. Is it clear for all? Clear. No doubts, right? Perfect. If no doubts, then uh, we have completed this part. So it is almost two past. It has passed two. So what we'll do is we'll wind up the session for the today. So next class, hopefully, what we'll do is I hope it will be an online session because due to this country situation now, so it will be also an online session. I guess this will be the same time, and inshallah next class we'll be starting gel electroporosis. We'll be completing probe hybridization, and inshallah I'll try to start cloning too. Right. So I hope this session was a useful one, and thank you for joining live. So let's wind up the class. Thank you for everyone. Right. So hope to revise what we have learned today, and if you prefer using that 147 technique, just try it. Give a try. I'm not composing. Just I'm not telling you you should do it, and I'm not focus forcing you all. right? It's your wish. uh, you can just try it, give it a try, 1 verse 1 technique, just 1.5 hour per day, and try it, right? And make sure to come revise this section next week, so it will be useful for you, right? Okay then, let's make it on the next week class. Thank you for everyone, and goodbye.